总共大概转移2000来人吧，根据排查的范围、排查的结果来有不断变化。没转移的那就尽量的封闭、尽量的隔离、尽量的取消校内公共，避免再出现进一步的传播。
坚定不移跟党走，为党和人民奋斗，是共青团的初心使命。100年来，在党的坚强领导下，共青团不忘初心，牢记使命，走在青年前列，组织引导一代又一代青年坚定信念，紧跟党走，为争取民族独立、人民解放和实现国家富强、人民富强贡献力量，谱写了中华民族伟大复兴进程中激昂的青春乐章。
Okay, class begin. Okay, sorry, morning. I'm making a mistake. I think our stereo class maybe quickly finished. Only left one chapter, one unit about the metabolisms of the nucleic acid. For this part, you can see how to synthesize the DNA, how to synthesize the RNA. This part we study next semester in the molecular biology. And this part, this semester, we only study the metabolism of the nucleotides. Nucleic acid, you can see hydrolysis and produce the nucleotides. So it's very easy. And hydrolysis and produce the nucleotides. How to synthesize RNA, DNA, next semester we studied, and today we focus on this part. You can see we study the proteins, the nucleic acids, vitamins, and so on, the structure, the function. And we quickly study the oxidative phosphorylation and the metabolism of the carbohydrate and the metabolism of the lipids. Amino acid. From today, we begin to study the metabolism of the nucleotides. Okay, this part divided the two parts. Referred to the purine and the pyrimidine. For the purine, you can see how to hydrolysis, how to cleavage, and another way how to synthesize the nucleotides. And for the pyrimidine, similarly, the hydrolysis and the synthesis. You can see divided two parts, and every parent nucleotides, you can say how to synthesize. Okay, nucleotides. This reviewed this part. We studied the structure of the nucleic acid. You can see nucleotides and the basic structure unit for the synthesize the nucleic acid. Nucleotides are synthesized in body. So unlike the amino acid, unlike the three kind of the fatty acid, all of these can be synthesized. So unlike the amino acid, nucleotides are not essential nutrients. Can be synthesized by myself. Okay, and another one you can see digestive absorption of nucleic acid. You can see from the food, you can see all kinds of the nucleic acid. You can see at the times you can see nucleic acid may be bonded with the protein, especially for the eukaryotic organism. Bonded together at that times you can see for human beings, you can see for hours, for all of us, the nuclear protein, you can see under the gastric acid condition and stopped itself. At that times, you can see the protein and the nucleic acid may be separate each other. And furthermore, the nucleic acid can be catalyzed and hydrolysis. Here, you can see the pancreatic nucleus act as the nucleic acid from the food. You can see produce the nucleotide. Steadily, nucleotides can't be absorbed into our body. At that time, you can see furthermore hydrolysis produced the nucleotides and phosphoric acid. And the nucleotides, you can see cleavage the glucosidic bond and produce the base, purine, the pyrimidine, and the pentose rubbers, deoxyribonucleic rubbers. So all of this you can see under the pancreas nucleus, under the pancreas nuclear studies, you can see can be hydrolyzed thoroughly, produced the base and the pentose. So and that times you can see the base and the pentose can be absorbed into our body. Pentose like the glucose, like the monosaccharide, can be absorbed into the mucosal cell, and base also can be absorbed into the body, into the mucosal cell. Okay, so as this part reviewed, you can see for the nucleotides, the function, you can see synthesize nucleic acid, many function and forms energy in vivo. For example, ATP. Another one, you can see participating in the metabolisms and the physiological regulation. For example, CAMP, cyclic AMP, act as the single message molecular. And third, you can see participating in act as the coenzyme. Here you can see NAD. The NAD you can see act as the coenzymes of the dehydrogenase. So, and another one, activated intermediate products.
Pakai juga kelas gua beda beku.
For example, you can see the synthesize the glucogen. The glucose molecule, you can see, must activated and produced the UDPG. So, and later we began to study the metabolisms of the two kinds of the base. For the nucleotides, is very easy. You can see the nuclear circuits catalyze directly produce the purine and the pyrimidine. You can see metabolisms of the purine nucleotides. Read this part, you can say AMP, GMP, the purine nucleotides, like this. Here you can see rubbers. Here is the adenosine. Here is the guanosine. Okay, for the first step, how to synthesize this? Rubbers come from where? From the PPP. From the pentose phosphate pathway. We studied the metabolisms of the carbohydrate. You can see the PPP. This pathway can be synthesized the rubbers. At that time, so you can see here for the pyrimidine, for the purine synthesized, you only focus on the base, focus on the purine. focus on the pyrimidine how to synthesize is okay. So okay, we studied the synthesize pathway. Here you can see Dylan synthesize and the standard synthesize pathway of the purine nucleotides. Maybe somebody think didn't know what means. What means? From the beginning to synthesize the purine nucleotides. You can say purine nucleotides was synthesized through a stereo of the enzymatic reaction. Applies several kinds of the simple molecules, such as the rubose phosphate, the amino acid, the carbon unit. Last times we studied, you can see here, carbon unit. Another one you can see is the carbon dioxide. All of this you can see synthesize the purine. The second method you can see is standard pathway synthesize purine. The purine nucleotides was synthesized using purine base or purine nucleosides through a simple reaction process. Maybe our body, the DNA molecule, you can see the RNA molecule, you can see hydrolysis and cleavage and produce the purine. Produce the purine stodic site, and again used again. Okay, we began to study the synthesis, the DNA synthesis of the purine nucleotides. Here you can see, except some kinds of the bacteria, almost all of the organisms can synthesize the purine nucleotides. For human being, for the mammals, you can see the liver is the main organ of the purine nucleotides synthesis. Following the small intestine and the thymus. Here you pay attention to can't synthesize in the brain and the bone barrier. Maybe some students begin to worry about it. In the brain, how to synthesize this? Only through the standard pathway synthesize. Here, this part, you must pay attention to. In the liver is very active. In the brain and the bone barrier. Can't synthesize through this pathway. Didn't synthesize the purine. Okay. For the purine, you can see how to synthesize the parent cycle. Here you can see the stable kind of the molecule. You can see the one carbon unit, the carbon 8, the nitrogen 9, glutamine, the nitrogen 3 comes from the glutamine, and the carbon 4, carbon 5, nitrogen 7 comes from glycine. The carbon 6 comes from the carbon dioxide, and the nitrogen 1 come from the aspartic acid, and the carbon 2 come from the one carbon unit. I marked with this number you pay attention to. This number we already studied in the nucleic acid. This chapter we studied the structure. We studied this part. Okay, how to synthesize from the number nitrogen 1, carbon 2, nitrogen 3, Yes or no, you pay attention to. How to synthesize this, you pay attention to. And first, how to synthesize, you can see the process. This process, my students know about this is okay. The thoroughly how to one step by step to synthesize the purine and pyrimidine. I think it is not needed. You only, but here my students pay attention to. This in the future maybe ask you. This purine cycle, every autumn come from where? You must remember. Okay, from here further the RMP, I know lodic monophosphate nucleotides, how to synthesize. Here you can see for purine nucleotides, refer to the RMP, how to synthesize. Another one, AMP and GMP, how to synthesize. Okay, you can see. Include the stable kind of the chemical reaction. First, you can see, based on the 5-phosphorus 1 purine phosphate PRPP. This molecule you can see act as the base and accept one by one group. One by one molecule you can see forms the purine cycle. Here you can see first since the cycle, this the first you can see provided there's the nitrogen. It is the nitrogen 9. Okay, you can see first you can see the 5-phosphorus 1 purine phosphate act as acceptor and catalyzed by the glutamine PRPP aminotransferase catalyze, you can see, and produces the 5-phosphobeta rubus amine. All in the starting the amino group transported to the PRPP and produced 5-phosphorus amine. And continue another molecule you can see, it is the glycine amide ribonucleotides synthesis catalyzed and one molecule glycine directly bonded to the amide group in the and furthermore you can see one carbon unit transported to the, you can see the glycine amide ribonucleotides, the transformase catalyze, and here bonded to the one carbon unit. This one carbon unit, it is a carbon dioxide provided. And furthermore, you can see another molecules glutamine provided the amino group and forms the formyls, glycine, amidine, ribonucleotides. And continue, you can see the FGAM, you can see the amino imidazole ribonucleotides synthesis. You can see, cyclase, kind of nice. First, you can see synthesize the 5 antin cycle and forms the 5 amino imidazole ribonucleotides. Maybe just some students think, why is the teacher, you remember the every antin, the location. Just you can say the PRPP act as the acceptor and acceptor the first amide group, the nitrogen 3, nitrogen 7, nitrogen 9. Where you remember the forms the glucosidic bond. It is for the purine bonded to the line. So you can just the PRPP accept the nitrogen. Here it is the line, nitrogen 9. And also you can see forms the 5 carbon. And furthermore, you can see and the GARR synthesis kind of nice. You can see accept the 1 carbon unit, 1 carbon unit. Here the it is the carbon 3. And furthermore, you can see the at the same time, you can see and furthermore, you can see 1 molecular carbon dioxide provided the 1 carbon. You can see carboxylase kind of nice. You can see produced the carboxy amino imidazole ribonucleotides. Synthesis kind of nice, and here carboxylase kind of nice. Produced the CARR. Maybe some students begin to think, you can see the from the ARR to the produced the CARR. These 2 parts, what different? What different? Maybe and first bonded the 6 carbon, O number 2. These 2 parts are the sequence. What is the right? You can see here. In fact, most times you can see carbon 6, carboxylase kind of nice, and synthesize this. And furthermore, you can see the multis kind of nice. The multis kind of nice can be from the N5 carboxy amino imidazole ribonucleotides can be changed to produce the carboxy amino imidazole nucleotides. If you bonded to the nitrogen 3, no worry, can be moved the location. And the last, first forms the carboxaminoimidazole ribonucleotides. And furthermore, you can see the another molecule, another aspartate provided the you can see furthermore, you can see one molecule the aspartate provided the amide group, you can see, and synthesize the social layers 5 amino imidazole 4 carboxamide ribonucleotides. And this molecule, you can see, furthermore, you can see the lysis catalyze and produce the fumarate, and sometimes you can see produce the 5 amino imidazole 4 carboxamide ribonucleotides. And furthermore, you can see added one molecule 4 mirrors group. You can see synthesize, you can see one molecule 4 mirrors transferase catalyze, you can see produce the 4 mirrors amino imidazole 4 carboxamide ribonucleotides. And furthermore, you can see here carbox group and another here amide group, you can see linked together and dehydration. produce the alcohol. Another thing can be used to produce the gluconeogenesis. Here you can say base connected with the carbohydrate. And another thing produces the carbon dioxide and the ammonia can be metabolism system to the amino acid can be produced the urease molecule. So for this, my students pay attention to statistic you can see and the uracil can be produced the acetylcholine. And for the styrene can be produced succinylcholine. At the same time, these two kinds of the molecular you can see can be produced the urease molecule. Okay, for this part, maybe quickly finished. And for this, try your best remember some more smaller logic point. And here, always ask you answer question. The two kinds of the synthesized pathway, please pay attention to. Another one. For example, this PPT you can see the statistic and the uracil metabolism and can be produced the carbon dioxide, ammonia, and at the same time it can be produced acetylcholine. Right. There's several kind of the smaller molecular, the K enzymes. Try your best to remember something. For the regulation, this part I is very easy. Explain something. You can say abide the main principle. The down products, it is the inhibitor. And the upstream products, you can say, act as the activator. It is all. So this part is okay. Maybe give you 2 days, you revisit all of the name contains, and Friday we take the last times class, and all kinds of the molecular you can say how to metabolism can be related to each other. So you studied this part. Reviewed all of the contents we already finished, and Friday we finished this subject. Okay, today we stopped here.
谢老师，谢谢老师。
OK 老师，谢谢老师。
谢谢老师。